I'm very happy to join you in this webinar today, everybody. My name is John, and I am the manager of Elabscience International Business. And for your information, just a little information, among Elabscience international markets, Malaysia is in the top 10. So today is our pleasure to have the opportunity to talk to our excellent researchers in Malaysia today. And our topic today is ELISA Kit. And ELISA Kit is the first and most fundamental product line since we started up our company. And in the next 45 minutes, my colleague, Vivian, our senior product manager for ELISA Kit, who has years of experience on ELISA Kit experiment, technical support service, and troubleshooting, will give us a detailed introduction on ELISA principle, process, operation, and finally troubleshooting. I'm sure everybody can find what you're interested in the presentation, so let's look forward to it. And next, I will hand it over to Lizzie, our regional sales manager for Malaysia, to give us a company introduction first. All right, Lizzie, here you go.
Yeah, thank you, John. Hello, nice to meet you all in this seminar. Let me introduce myself again. My name is Lizzie. I'm from the International Business Department of Science. I'm in charge of the market. Okay, so today our webinar is entitled as ELISA Experiment Guide from Novice to Master. It is our honor to introduce you our star product in Malaysia. Actually, this is also the first time we carry out such a webinar for ELISA Kits. Okay, in the next slide, I'll share with you the agenda for the webinar. In the beginning, some of you might have used our ELISA Kit or might have heard of our company Elabscience. Well, someone might haven't. So in the beginning, I'll introduce our company very briefly. In the second part, I'll hand over to our speaker today, Vivian, for the introduction of ELISA introduction. Okay. The third part is exciting part is our quiz time. We have set up 5 questions, so all the winners can get our gifts. The last part is FAQ. So you can type your questions during our presentations. Our technical support online will respond timely. Okay, so here we go. For our company, Elabscience, founded in 2009, is headquartered in Optical Valley Medical Park with more than 500 employees so far. Our products mainly covers immunology-related products, including ELISA Kits, antibodies, proteins, as well as other assay kits. In addition, we can also provide custom services for your research. And in the next slide, I'll show you all our main product lines. ELISA Kits, so far we can provide more than 4,200 ELISA kits and 1,900 clear kits. In this year, we have released our new product line, ELISA one-step ELISA series gradually. This ELISA kit can shorten your experiment time to only 19 minutes. Besides ELISA kits, we can also provide the metabolism assay kits, cell functional assay kits, as well as 12,000 primary antibodies and secondary antibodies. And the good news is, from this year, we resumed the supply of cell lines and cell culture mediums. So if you have any requests for our products or you want to know more about our company, you can contact our authorized distributor, Apical Scientific. Okay. In the next slide, I can show you some pictures about our company. It includes labs equipment and facilities. You can see our animal house as well as the GMP plants. And you can also see our facilities like the flow cytometer, liquid nitrogen tank, as well as the fluorescence microperimeter. So far you might have a little expression about our company Elabscience. In the next part, I'll hand over to Vivian for the introduction of ELISA experiments. Okay, Vivian.
Okay, hello everyone, nice to meet you. And let me introduce myself again. My name is Vivian and I'm from the ELISA pipeline of Elabscience. It's my pleasure here to share you with my knowledge and my experience on the ELISA methods. And our topics today are divided into 3 parts: the principle of ELISA, the operation process, as well as the data analysis, and the ELISA frequently asked questions. First of all, let's take a look at what's ELISA and the comparison between the ELISA kits. ELISA is a highly sensitive experimental technique to detect the content of the target based on the immune reactions, and it has 4 bases. The first one, the capture antigen or the antibody need to be coated on the solid phase carrier and also called the microplate. The second is the immobilization of the antibody and the antigens. The third is the enzyme-conjugated antibodies and antigens and the substrate, namely the chromogenic agent. And the last one is the chemical signals is converted to the electrical signals by detecting the molecules, and the target protein is quantitatively analyzed by the OD value or the luminescence value. And from the principle of the ELISA, we can tell the ELISA products have 4 characteristics. The first is the specificity. Based on the complementary relationship between the chemical structure and the spatial configuration of the antibody and the antigen, the reaction of them has a very high degree specificity. And the second is the quantification. After adding an increasing amount of antigen to a constant amount of the antibody to form an immune complex in the most suitable range, that is, the equivalence band, will show the equal proportionality and allow the analyte to be quantified. The third is the sensitivity. ELISA uses the enzyme conjugate and the built-in evidence system to make the sensitivity of the product reach the level of the micrograms or even the picograms. And the last is the high efficiency and the economy. ELISA requires small volume to detect large numbers of the samples, and the experiment time can be reduced to only 80 minutes. And based on the above 4 characteristics, ELISA are widely used in scientific research. In order to meet the needs of the market and the customers, we are constantly optimizing our products and launch the new ELISA series. Here I have listed the ELISA products series of Elabscience. The traditional clear and the ELISA kits is our traditional series, which is furthered by the numerous items and the high performance. Quickie Pro ELISA kits is the upgraded series. The whole experiment takes only 90 minutes, which greatly help with the experiment efficiency. And the MS ELISA kits are specially designed for the mouse item detection, which can effectively meet the needs of the small sample size of the mouse by reducing the value to only 35 microliter or 25 microliter. And the SARS-CoV-2 ELISA kit is our new series, and it contains a full range of COVID-19 related test kits that can be used for its research and the prevention. Since the Quickie Pro ELISA kit is the new series, how is it different from the traditional ELISA kits? And traditional ELISA kits is also called the 3-step ELISA kits, and that means the samples, the built-in latent antibodies, and the enzyme conjugates are added in 3 steps. However, the QuickPro ELISA kits manage to add the samples and the HRP conjugate at the same time to complete the entire experiment with only 1 step. With the reduction of operation steps, the total assay time can be reduced by 1 to 2 hours, and the loading value of samples has also been reduced by 50 microliters per well. Okay, after the introduction of the principle of the ELISA kits, now let's move on to the main part today, the operation tips and the tricks. Please feel free to type your questions in the chat window during my presentation, and we will answer the questions one by one online later. And this part, we focus on sharing 3 aspects, including the sample processing, operation procedures, and the data processing. First, let's take a look at the sample processing. The common sample types used in ELISA experiments include the serum, plasma, cell supernatant, cell lysates, tissue homogenates, as well as some other biological fluids such as the saliva, urine, milk, and so on. Different sample types require different preparation method, and the good quality samples is the first step to ensure that the accuracy of test results. First, let's look at the processing of the serum samples. The supernatant obtained from the coagulated whole blood by centrifugation is known as the serum. We can choose a vacuum tube without the anticoagulant or with the coagulant to prepare the serum, and during this process, the fibrinogens and some coagulant factors are removed. The second sample type is the plasma. Plasma is derived from the supernatant of the unclotted whole blood with the anticoagulant. It contains the fibrinogens and the anticoagulants, and some other components are equivalent to the serum. The common anticoagulants used in this process include the EDTA, heparin, the sodium citrate, and among them, EDTA is the most recommended anticoagulant. Therefore, only plasma samples can be used for the fibrinogens and the anticoagulant-related factors detection, since they are involved in the blood clotting. But for the conventional secretory proteins, either the serum and the plasma can be selected as the appropriate sample type. Although the preparation of serum or the plasma samples is fairly simple, however, to obtain the ideal samples, the following points should be noted. The first one, we need to avoid the bacterial contamination. The false positive results are always caused by the endogenous enzymes contained in the bacteria or the secrete during the bacterial growth. And the second, avoid hemolysis and the hyperemia. Hemolyzed or the high-fat samples can cause the non-specific color reactions in the ELISA experiments and resulting in the inaccurate test results. And thirdly, we need to ensure that samples are clarified and homogenized before use. And the last point, samples should be assayed be tested as soon as possible after collecting, or aliquot or freeze them for further use. Avoid repeated freezing and thaw cycles. And before the experiment, it's recommended to centrifuge melted samples again to remove the sediments. The third sample type is the tissue homogenates. The main point of this sample type is that the tissue blocks should be minced and fully ground under the ice-based condition. In order to further lyse the cells, It's recommended to introduce the repeat freeze and thaw operation. The specific operation of this step is that store the homogeneous at -80° for 1 hour, or the liquid nitrogen for half an hour, then place in a 30° water bath with the slightly shaking to melt quickly. And this operation can be repeated 1 to 2 times to get the cell lysis. And for the special samples or the samples with lower target protein, the grinding radius can be reduced moderately. The fourth sample type is the cell culture supernatant. Cell supernatant samples could be prepared by simple centrifugation step to remove the cell debris and the magazines. Cells can be cultured in different well plates, but we should make sure that the numbers of cells in each group is basically the same and the growth state is fairly good. In addition, it's desirable that the cell culture media does not contain the serum components. The fifth sample type is the cell lysis. For adherent cells, the cells should be separated by the trypsinization and then collected by the centrifugation as in the suspension cells. Then the cells were resuspended to prepare the cell lysis by the repeated freezing and thawing or the ultrasonic method. If the numbers of the growth state of the cells varies greatly between the wells, it's recommended to correct the data by measuring the total protein concentration of each well. By the way, all of these common samples, such as the serum, plasma, tissue homogeneous, or the cell samples, are suggested to be used within 7 days when stored at 2 to 8°, or otherwise the samples must be alcoholed and stored at -20° for no more than 1 month, or -80° for no more than 3 months, and avoid repeated freeze and thaw cycles. After obtaining a good sample, we need to prepare the instruments and the reagents required for the ELISA experiments, and you can see in the picture. Then we could design the experimental scheme. Usually we tend to set the 2 columns of the plate as the standard wells, containing 8 gradient concentrations with the duplication. The standard working solution is added to the plate from the bottom to the top and from the low concentration to the high concentration. And the last 10 rows of the micro well strips are the spotting wells for the samples to be tested, which are designed according to the experimental needs. Generally, it's not suggested to set the duplicate. It's also suggested to set the duplicate wells, including the normal group, the model group, and the treatment group, referring to the experimental requirements. After the design of the experiment, we need to carry out the preliminary tests to verify whether the samples is suitable for the normal experiment. At the same time, irreverent variables are also precisely controlled in these pre-tests, and the sample dilution ratio and the optimal experimental conditions are further determined. It also avoids the waste of the energy, material, and the financial resources caused by the blind experiments. And note, if the sample dilution factor is too high, it's recommended to dilute the sample step by step, and the maximum dilution cannot be more than 100-fold. Okay, then let's focus on the specific operation step in the ELISA assay. This part mainly includes the 4 processes: the sample loading, washing, color rendering, and the color remetry. We have put our ELISA operation videos of both the sandwich and competitive method online. And for details, you can log in our Elabscience official website to watch the videos. And the ELISA kits of Elabscience are mainly based on the sandwich and the competitive method. And let's take a closer look at the procedures for both of these two kinds of kits. The difference between them is that the sample and the built-in latent working solution are added in two steps in the sandwich method, where in competitive method, these two reagents are added at the same time. The volume of both two reagents are also being reduced by 50 microliters, and the incubation time is also shortened to 45 minutes with the competitive method. Although the competitive method have the advantage of the ease of operation and the small sample size, however, the sandwich method can produce more sensitive kits to meet the detection of the low concentration items. Therefore, both methods are widely used in the scientific research. Next, I will explain more on the ELISA operation procedures. The first is the operation of the adding step. Standards are suggested to be added in order from the low concentration to the high concentration, and the same concentration need to be added in parallel. And for the samples, we must pay attention to change the pipette tips in time. And before the adding process, it's necessary to mix the reagents well and avoid air bubbles. All the solutions should be balanced to room temperature before use. And then suspending the pipette vertically to add samples to prevent the pipette tip from touching the wall or the bottom of the well. And at the same time, it's also recommended that the sample addition time should be controlled within 10 minutes to ensure the consistent original reaction rate between the different wells. The second step is the incubation. And before the incubation operation, it's necessary to check whether there are omissions in the last addition steps. And if not, timely adhere a suitable size sealer on the ELISA plate and then mark it accordingly. Horizontally move the plate to the 37 incubator without overlap placement. And note, during this process, the incubation temperature, the incubation time, and the color development time must be strictly controlled, and a new sealer needs to be placed for each incubation step. The third step is the washing operation. Washing is a crucial step in the ELISA assay, and any improper operation will affect the final results. After taking out the ELISA plate, quickly and vertically shake off the liquid in the well and pat it dry on a clean absorbent paper. It's recommended to replace the paper each time, and when the plate is clamped. And then on the washing operation, we can choose the manual operation or with a plate washer. The washing step is repeated 3 or 5 times according to the manual. And note, during this process, the washing buffer should be prepared and used without contamination. And at the same time, we also need to pay attention to control the volume and the soaking time of the washing buffer. The last step is the color rendering and the colorimetry. Color development is the final incubation step in an ELISA experiment. At this point, the enzyme catalyzes the colorless substrate to produce a colored product. The color reaction should be carried out in the dark, and the reaction temperature and time should be controlled according to the manual and strictly to be accurate. Colorimetry is the last step in the entire experimental operation. After adding the stop solution to terminate the reaction, use a microplate reader to measure the optical density of each well at a wavelength of 450 nanometer. And before the sampling before sample detection, the microplate should be turned on 15 minutes in advance to warm up, and the parameters are set completely. At the same time, pay special attention to the bottom of the ELISA kit, bottom of the ELISA plate, and ensure that it is dry and clean. Okay, after the chemical signals are converted into the electrical signals by the instrument, we need to perform the data analysis operations. The 4-parameter logistic function is the most suitable equation for the ELISA curve fitting, and any software that can use to fit this equation would be applicable. We always choose the orange software to fit the curve and enter the standard data into the orange with the standard concentration value in the X column and the corresponding optical value in the Y column, and select the 4-parameter logistic function for the long liner fitting to obtain the standard curve. The OD value of the samples to be tested is brought into the curve to calculate its concentration value. If the samples to be tested is diluted, the final value needs to be multiplied by the dilution factor. And more data analysis details can be found in the technique article on our website. And through the software fitting, we will get the standard curve shown in the figure. Among them, the experimental result of the sandwich method is that the concentration of the standard is proportional to the optical density value, while in competitive method, the concentration is inversely proportional to the OD value. And above is all about ELISA operation tips and tricks, and hoping that you all could master the accurate sample processing and operation method to get the ideal test results. And by the way, if the abnormal results such as the low signal per CV or other unexpected data still occur, you all could analyze the causes of the problems one by one through the ELISA technical guides we have compiled, or directly contact our technical support to solve the problems and given the main detail of the ELISA operations. Okay, let's come to the excited part, the quiz time, and I will transfer the lead to Lizzie.
Yeah, Vivian, thanks for the practical tips and tricks for ELISA experiments. So let's take a break for the exciting quiz time. We have set up 5 quiz questions closely related to Vivian's presentation. So once the quiz starts, you just need to type your answers in the chat box, and we will pick the winners for each quiz. And all winners can contact Miss Jenny or contact our online technical support for the gifts. Okay, so let's start.
Okay, quiz 1. What are the differences of one-step Quickie Pro ELISA kits over the traditional ELISA kits? And it's a multiple choice.
Yes, multiple choice means the answers is more than one options.
Yeah.
Please type your answers all together. It's a multiple choices.
Multiple choice.
All right. Let's see, there are some answers already.
Okay. Oh.
Okay. Yola.
We see no winners. Okay. Okay. Vivian, can you tell us the correct answer?
Okay. A, B, D are correct. And more ELISA items is the strength of our traditional ELISA kits. and it contains the highest numbers of items among all of our ELISA kits, and the other strength is our one-step quickie pro ELISA kits.
Yeah, Jenny, can you take a note for the winners? For the winners, please leave your email box or your contact number to Jenny.
Yeah.
Okay. So
Thanks. I will text them.
Quiz 1. We'll enter the quiz 2 now.
Okay. What is the difference between the serum and the plasma?
Yes, this quiz is very simple because it only had 2 options.
Okay. Yeah. All the answers are correct.
Okay, so
yeah, A is correct. Unclotted blood will yield plasma after the centrifugation, and it contains coagulation factors.
Yeah, please contact Jenny for the gifts.
Okay. And quiz 3. What are the storage conditions for the ELISA test samples? It's also multiple choices.
Yes, this part is included in the sample preparation method, right? Vivian?
Yeah, yes.
Okay. And the winner appears?
Appears.
Okay. Okay.
Okay. A, C, D are correct. And the premise of valid result is the good sample quality. And therefore it's not recommended to store the samples at 2 to 8 degrees for more than 1 week.
Yeah. So that's all for the quiz 3. There are remaining 2 quizzes. So if you haven't got the gifts, please participate in our rest 2 quiz questions. Okay, so let's move on.
Quiz 4. What are the operation points included in the ELISA experiment? It's also a multiple choice.
Okay.
And it's an easy quiz.
Yes, very easy. This content is included in our content part, right?
Yes.
All right, there are already some correct answers.
Yeah, all the choices are correct. Although ELISA is a relatively conventional and simple experiment, it will be affected by many factors because it's a micro reagent reaction. Therefore, for ideal results, every step and every operation details in the ELISA experiment is worth paying attention to. Yeah.
Okay, as Jenny also graduate the winners, so thank you.
Okay, quiz 5. What's the relationship between the standard solution concentration and the OD value of the sandwich method and the competitive method? It's also a multiple choice.
Yes, that means the answer is the mono 1 options. Yeah, it's a very basic content for the ELISA experiment.
Yeah.
Right. And the correct answer, yes, everybody got the correct answer.
Yeah, A and D are correct. The sandwich method shows the direct proportion in the cup, while the competitive method shows the inverse proportion.
Okay, so that's all for the quiz. Congratulations to all the winners. Please contact Jenny for the gifts.
Okay, let's move on to the last part today. ELISA frequently asked questions. Welcome to our sales manager, June, to hold this part, okay?
All right, so nice to talk to you again. Well, we have received a lot of questions from our customers in our daily work. So now let's see some of the questions that are most frequently asked. And let's see. Okay, this is the most commonly asked questions. Question number 1 from our customer. Given the size of the serum and plasma samples, how can you calculate the volume of the whole blood samples?
About 350 microliter of the serum or the plasma samples can be prepared from the 1 milliliter of the whole blood. Therefore, it is advisable to collect the whole blood with more than threefold the volume of the expected serum and the plasma.
All right, thank you, Vivian. This is the most commonly asked question.
Yeah.
Then let's see question number 2. How many samples can a 96 test kit detect?
For 96 test kits, run up to 88 samples if the standards and the samples are assayed in the simple well, and run up to 40 samples if both the standards and the samples are assayed in duplicate. And for each test kit, it's necessary to test the samples together with the standards. In addition, it's recommended that all the standards and the samples be assayed in duplicate to improve the accuracy of the test results. However, the duplication is selected according to the experimental design and without the rigid requirements.
All right, thank you. This is really important before we start the experiment. Then let's see question number 3. Can the dissolved standard still be used?
Generally, it's not recommended to use the dissolved standards, and the 2 vials of the standard power in each kit will be sufficient to complete the experiment. But if the end user does have the need for several assays and with the standards, please add coat and store the dissolved standard -20 degrees and avoid repeated freeze and thaw cycles. The freeze standards would be valid within 2 weeks.
All right, thank you. Then let's see question number 4. When the sample or reagent size is insufficient, can all reagents be reduced in equal proportion to carry out the experiment?
No, the system of all pre-coated or the ready-to-use kits is optimized and fixed. The volume adjustment of the individual or the overall components may lead to the abnormal results. Therefore, it's recommended to arrange the experiment according to the actual volume of the reagents. In addition, do not use the other source or the different batches of the reagent to meet the volume shortage problem. And if the sample volume is insufficient, it's recommended to consult the literature or contact the technical support to get the sample size information. It's very important to know that the detection range of the kits is not equal to the concentration range of the item in the sample, and the loading volume of samples is also not equal to the actual demand. And many items could be diluted to reduce the sample values since the kits are very sensitive.
All right, then let's see the next question, question number 5 from our customer. Should the color development time be strictly controlled at 15 minutes?
The color development time was determined according to the blue gradient of the standard. When the 4 low or high concentration standards shows a clear blue gradient, and the reaction could be terminated by the stop solution. And the color development time is usually controlled within 5 to 30 minutes.
All right, then let's see the last commonly asked question, question number 6. Can the standard curve be fitted by Excel?
No. Although the Excel is the most common scale fitting tool, it's not recommended to use it to fit the ELISA standard curves, since the common fitting method of it, such as the linear fitting and the polynomial fitting, are not effective in reflecting the response curves of the antibody and the antigens. And our lab usually use the orange software and fits the 4PL function to get the ELISA results. Other software such as PASS or the graph pad, which could also fit the 4-pair cups, is also suitable.